Hi, I'm Rachel Jabali, an assistant professor of botany at Colorado College. I teach field botany, amongst other things, and I'm so fortunate to work with class after class of students eager to learn about our native flora. It is my absolute pleasure to introduce one of my former students today, our speaker, Piper Bodart. Piper is originally from Northern Illinois, and we first met when she was a student in my introductory biology of plants class. She was exceptionally bright, and I was excited to learn about her deep interest in lichens, not a common thing to hear from a freshman. Apparently, she'd even written part of her college admissions essay about them. My research focuses on flowering plants in the Andes and Australia, and I tried to get her to pivot to one of those projects, but she stayed firm in her conviction that lichens were what she most wanted to research, and she wanted to get out into the field here in Colorado. She found out about a region of the Flat Tops wilderness that had been surveyed repeatedly by some of the nation's preeminent lichenologists at semi-regular intervals, but no recent follow-up had been done. She developed all of the necessary contacts and permissions to be the one to do that work. An absolute high point of my summer last year was when our entire lab got to accompany Piper to the Flat Tops for her field work. Piper knows her stuff and was very efficient at doing her work and also very accommodating, teaching her peers all that she knew. She then went on to deposit her lichen specimens at the CU Herbarium and the Smithsonian, where her collections can inform all types of scientific studies for generations to come. It certainly would have been more than enough for an undergraduate thesis project to collect and identify these lichens, but Piper went much further. We worked with Lauren Trotta, a PhD candidate at the University of Florida, on applying a cutting-edge approach, community phylogenetics, to understand the interplay of ecological and evolutionary forces shaping the lichen community. Scientific community was mightily impressed when Piper presented her work at the Botanical Society of America meeting last year. Piper completed her thesis with distinction and graduated summa cum laude with a degree in organismal biology and ecology from Colorado College this past May. She earned the department's Richard G. and Reba Beidelman Award, given to the graduate with the greatest potential for becoming a professional ecologist or field biologist. Obviously, Piper has made her faculty very, very proud. She now resides in Phoenix, where she works for Moon Valley Nurseries. Piper has recently started volunteering with lichenologist Dr. Frank Boongartz at the Arizona State University Herbarium on a lichen databasing project. Join me in a virtual welcome to Piper, who will now present her talk, Common Lichens of the Flat Top Wilderness Area, Biology, Identification, and Research Applications.
As Jabali said, I just wrapped up my final year at Colorado College, including my thesis project on lichens. So today I wanted to take this opportunity to talk to you not only about the research that I did, but more broadly about my study subject of lichens, because lichens are a study subject that kind of falls through the cracks in some of your typical biology classrooms. It's not an animal, it's not a plant in the traditional sense. So sometimes it's not a subject that's really well covered. So today I wanted to go over the basics of lichen biology, lichen morphology, and a lot of the characteristics that are important to know when trying to recognize and identify lichens in the field. After that, I'll transition to talk more about the lichens that I worked with in particular in the flat tops and some of the conclusions of my research. But before we get into that, if you all feel comfortable typing up a sentence or two about your experience with lichens, maybe what you know, what your interests are, that way we can just start off the conversation seeing what you know and what you hope to learn today. While you type that up, I would say that when I came to Colorado College, as Jabali said, I had this deep curiosity about lichens. I knew that I loved taking pictures of them. I loved their little details, but let me tell you, I did not know what I was getting myself into when I decided to take on a whole senior thesis about this symbiotic organism. I would soon find out that lichens are very complex, and they come with this dictionary of their own with really dense terms that I had never come across before in all my biology studies. So hopefully a lot of you were able to get an email from Maggie, including just a quick list of some lichen terminology that covers a lot of the definitions of the terms I'll be using today. That's a really good resource when using these words that aren't really common sense, and hopefully you can keep that list in mind going forward as you choose to learn more about lichens. So we'll just take a quick look at some of the comments just to see where everyone is at coming from today. So it looks like we have some beginners wanting to learn more. Beginning workshops from Dr. Tripp. She's a great resource up at the University of Colorado Boulder. Wonderful, so some knowledge about growth forms, which we'll go over today. Some people studying lichens in Southwest New Mexico. So this is fantastic, a really broad range of experience. So hopefully today everyone can take something away from the talk and bring with them going forward as they learn more about lichens. So we can go ahead and get into it. And first, I will say if the videos are in your way, they might cover up portions of the slides. So feel free to minimize the videos. You should be able to do that on your end. So the question that I always start with when talking about lichens is, what is a lichen? And this is really a fantastic question to ask because lichens are not a single organism. Rather, they are two or more biological components living together in symbiosis. So this symbiosis is still being actively studied. It's not entirely understood. There's more components that are emerging. People are trying to figure out exactly which roles all these components play, but every lichen will contain a mycobiont and a photobiont. So the mycobiont is the fungal portion of the lichen. So this is the main component of the lichen that you see when you're looking at an individual, and it comprises both the internal and the external structure of that lichen. So the role of the fungus in this symbiosis is to provide a suitable microhabitat for the algal partner. So what's interesting about a lot of these algae is that they are not able to survive in the habitats where lichens grow on their own. You're not going to find them free living in those environments. Rather, they need the structure of the fungus to protect them from UV radiation and the threat of desiccation. Additionally, the fungus is responsible for acquiring mineral nutrients from the substrate. So as it interacts with the substrate it's attached to, starts to break it down a little bit, that frees up mineral nutrients that are then accessible to both the fungus and the algae. So this fungus comes from a really diverse subset of fungi belonging to the phylum Ascomycota. And these fungi are united by the fact that they have sap-like asci in which they produce their spores. And so there are over 19,000 species of fungi that are able to interact in this lichen symbiosis. So that's incredibly diverse. There's nearly 1,000 genera of these lichens. And what's cool to me is that this was not singularly a one-time evolutionary event. Rather, this process of lichenization occurred at several different time points in several different clades across evolutionary time. So this is really a partnership that has withstood the test of evolution and is continuing to diversify today. So it's important to note that the lichen taxonomy is based on the fungal portion only. So when you're looking at species names, phylogenies, and genetic sequencing, all of that is done on the fungus rather than the algae. When we take a look at the photobiont, which is a photosynthetic algae, it comes from a much less diverse subset of organisms. So either 25 genera of green algae or 12 genera of cyanobacteria or blue-green algae. So as the name suggests, the primary role of this algae is to produce carbohydrates via photosynthesis that it then shares with the fungus in the form of different sugars. What I find cool about the photobiont is that you can actually distinguish it visually. So if you take a cross-section of a lichen and then look at it with your hand lens, you'll either see algal cells that are bright green in color, which would be the green algae, or a darker black, blue-green color, which would be a cyanobacteria. So we'll go ahead and take a look at how these two symbionts come together to form a lichen. So the body of a lichen is called the thallus, which is simply the vegetative, non-reproductive portion of the lichen. So the thallus has several main components, including an upper cortex, which is composed of densely packed fungal hyphae or fungal filaments. Below that, you have the algal layer, in which the photobiont cells are loosely interwoven with fungal hyphae. And then finally, you have the medulla. So when you look at a cross-section of the lichen, the medulla will often take up the largest portion of that cross-section. And it consists of loosely packed fungal hyphae that often appear white and cottony when you look at them. And so this exact structure can vary depending on the growth form of the lichen. So there are three primary growth forms. These include the foliose, fruticose, and crustose species. Some intermediates exist between these. We'll look at those as well, but we'll start off with the foliose lichens, which are perhaps the ones that you imagine when you think of lichens you've seen on rock surfaces or trees that you've encountered. And these are the lichens that have a flattened leaf-like thallus that has a distinct upper and lower surface. So oftentimes you'll note that the upper side of the lichen is actually a different color from the lower surface. And the lower surface may have a well-developed cortex, or that cortex might be lacking. It depends on the species. The foliose lichens can attach to their substrate in a couple of different ways. So rhizonate foliose lichens attach to their substrate via structures called rhizemes or rhizoidal hyphae. So rhizemes superficially resemble roots. So there are these stringy little structures that attach to the substrate, but it's important to note they don't serve any vascular function like a root does. They're not uptaking water from the substrate and distributing it throughout the thallus. They are simply a means of attachment. So these rhizonate lichens are the most complex out of all the lichen growth forms, meaning that they can sustain growth up to a ½ meter in diameter. When we look at umbilicate foliose lichens, they attach to their substrate at a single point instead of many different points, and the singular holdfast is called an umbilicate. So a great example of an umbilicate species is the genus Umbilicaria, pictured on top. And then other common genera of foliose lichens that you'll likely come across in Colorado include Xanthoparmelia on the lower left and Physia on the lower right. So moving into the fruticose lichens, this growth form is characterized by its three-dimensional growth. So these lichens either grow upwards like a stalk, or they'll grow downwards like a pendant. What's unique about these lichens is that they will often have a singular cortex that envelops their entire thallus. So their thallus is often cylindrical in shape. Similar to the umbilicate lichens, the fruticose lichens will generally attach to their substrate at a single central holdfast. And since they have such long growth, they require additional internal support structures to sustain that growth. So these include either a central skeletal cord of hyphae or a hollowed out center part of the cylinder so that they're light enough to sustain that growth. So some of the genera that you are likely to see in Colorado include Usmia, pictured on top, and Clidonia. So together, the foliose and fruticose lichens comprise the set of macrolichens, which are called macrolichens simply because they have generally larger thalli and larger features. So looking into the microlichens, we have the crustose growth form, whose entire lower surface grows in direct contact with the substrate below it. So these lichens do not have a lower cortex. Instead, the hyphae or the filaments in their medulla adhere directly to the surface below them. So oftentimes you'll see many individuals of crustose lichens growing together in one indiscernible patch, and they are impossible to separate from their substrate intact in one piece. So when you collect crustose lichens, you have to take the whole substrate with you. A feature called a prothallus may or may not be present on a crustose lichen, and a prothallus is simply a mat of purely fungal tissue that grows below and around a crustose lichen. And since it's fungal only, it doesn't contain any photobiont cells, so that means it will be a different color than the rest of the lichen thallus. So that's a feature that is good to be able to recognize when trying to identify a crustose lichen. So the thalli of these lichens may be smooth and continuous in just one piece, or it may be composed of many small scales or areoles. And some interesting subsets of crustose lichens include endoploidic and endolithic lichens. So endoploidic lichens are those that grow with their thallus embedded in the upper surface of bark or wood, such that you can't really see the thallus when you're just looking at that substrate. And similarly, endolithic lichens grow with their thallus embedded in the upper layer of rock surfaces. So some common genera of Crustocyclines in Colorado include Rhizocton, pictured up left, Candelariella, and then Licidea. So you're likely to come across those on your botanizing hikes. Next, we'll take a look at the squamulose growth form, which is another microlichen, but it is an intermediate between the foliose and crustose growth forms. So similar to a crustose lichen, the squamulose lichens lack a lower cortex, and they're composed of small scales. They can either attach to their substrate along the entire lower surface or along the thallus margin, which you can see pretty clearly in this lower image. And when they do that, they do resemble foliose lichens. So when these little scales align like shingles on a roof, the lichen is described as imbricate. Again, there's just a lot of terms peppered in here that are good to know when trying to learn terminology for lichen identification. So some of the common genera in Colorado include Sora and Clidonia. You may have noticed that Clidonia is an example of growth of Fruticos and a Squamulose lichen, and that's because it's a unique genus in that it has both a primary and a secondary thallus. So the primary thallus is the one that grows low to the ground or close to the substrate, and it's a squamulose form. And then growing upwards out of that, you have a fruticose thallus, that's the secondary thallus. So that's a really cool combination of features that is seen in Clidonia, making it a pretty easily recognized genus in Colorado. Next, let's take a look at the different lichen substrates. So substrates are simply the surface that a lichen is growing attached to. There's, of course, a bunch of different terms describing these because everything in lichenology comes with a lot of terms, but some of the most common ones that are good to know for identification include Sacchlids lichens growing on rock, lignicolous lichens growing on wood, and so on. So again, just a couple of terms that are helpful to know when trying to narrow down what lichen you're looking at, because sometimes two lichens that look very similar, one species will be known to grow on tree bark, one species will be known to only grow on rock, so that can help you distinguish between species. Also helpful to begin distinguishing between species are the sexual structures that lichens produce. So only the fungal portion produces sexual structures that are visible when looking at a lichen. The first of these being Apothecia. So Apothecia are the disc or cup-shaped fruiting bodies of the fungal component. They include a spore-producing layer that is exposed on the upper surface. So if you look at these images, the upper left image has Apothecia that have a spore-producing layer that is this peachy salmon color. So looking at that, that is only fungal. There's no algae in that portion of the lichen. And this is generally round in shape. If the Apothecia are elongated, then they are described as lyraline. And a really important feature that distinguishes between Apothecia are their margins. So you can either have a lecanorine or a lecidine margin. On the left, we see examples of lecanorine margins. So around each Apothecia, you see a ring of tissue that is the same color as the lichen thallus. And so this ring around the Apothecia contains photobiont cells and is therefore described as lecanorine. If we look over on the right side of the screen, there are black Apothecia in both of these species. You can notice that these have lecidine margins because there's no ring of lichenized tissue around them. It is simply a black Apothecia and no ring around them. So again, some good terms to know. There's another sexual structure that lichen fungi produce called Parathesia. So instead of being an open cup shape, these instead are flask-shaped fruiting bodies. And you can't see the spore-producing layer. So there's no bright colored layer exposed in Parathesia. And the spores are simply released through an opening called an ostiole. So these are not as fun to take photos of, so I don't have those available in these slides, but they are, again, an important feature when identifying lichens. They're just not as conspicuous or easy to identify using the hand lens in the field. They generally look more like black warts or pimples embedded in the lichen surface. Other features that are important to be able to recognize on a lichen are the asexual. So the sexual reproduction of lichens is a pretty tricky process because if you need two organisms living together successfully to create this symbiosis, but you're releasing fungal spores into the environment and then hoping that algal cells somehow find those spores and enter a partnership, that's just not a very reliable way to ensure the further population of a lichen. So instead, a lot of lichens will also participate in asexual reproduction through a couple of different features. So the first being spiridia, which are spherical in shape. They can be granular to flowery, but essentially they are just a cluster of photobiont cells which are enclosed in a layer of fungal hyphae. So they sit like spheres on the top of a lichen surface, either all throughout the thallus or in distinct pockets along the margins. So these are very small features, around 20 to 100 micrometers in diameter, but you can see them with a hand lens, and they're a good feature to be able to recognize in the field, as well as isidia. So isidia are small outgrowths of the thallus, so they attach at the base of the lichen, the thallus, and they are cylindrical in shape. They can be branched or bulbous. And again, they are pretty small features that you can see with a hand lens that are important for lichen identification. So we'll take a moment now to pause and see if there's any questions on the first portion of this talk, including the lichen symbiosis, morphology, and so on. So if there's any questions, I'll just keep an eye out for those in the chat box. And we will also have some time for questions at the end. So feel free to either ask anything now, or we can also go over it at the end of the presentation. All right. So then we'll go ahead and transition into the second portion of the talk, which focuses more on the lichens of the Flat Tops Wilderness area in particular. So the Flat Tops Wilderness area is located in the northwestern portion of Colorado in Garfield County, and it's distributed between the White River and Route National Forest. So it's situated at an elevation of 10,500 to 11,500 feet. And it's characterized by these beautiful flat topped plateaus with rolling alpine meadows and lakes in between. So it's a gorgeous study site. And I chose to work here because it has a history of lichen monitoring. So in 1982, the Forest Service initiated a program to monitor air pollution using lichens as bioindicators. So lichens are a really great tool for biomonitoring because there are some species that are really sensitive to low levels of air pollutants. So essentially, if you note that there are healthy, stable populations of these sensitive species initially, but over time they start to dwindle or decline, then that's a good way to indicate that air pollution is increasing. Similarly, lichens can bioaccumulate certain elements that are common in air pollution. So if you sample the thallus of a lichen, you can actually collect information on the quantity of certain pollutants present over time periods. So this program was initiated in 1982 by lichenologist Mason E. Hale, and he essentially set up the baseline data for this study. So he inventoried 21 study sites across the flat tops, creating species lists for each site. And then he also established 12 permanent photo quadrats to analyze lichen change over time on a smaller, more specific scale. So each of these quadrats is only 8.5 by 11 inches in size. So it's a really small section of rock, and they're all placed on rock surfaces. So it's really just analyzing Sacchlids lichen communities. And what's cool about having photographs and comprehensive species lists is that you can track the changes of individual lichens over time, especially when you start to do these repeated inventories that Jabali had mentioned. So The inventories were repeated again in 1992 by a group of lichenologists called Lichens Unlimited, and then again in 2009 by a group of lichenologists who are now based out of Brigham Young University in Utah. And so what this does is set up this structure in which we have lichen inventories in specific quadrats that have timestamps on them. So we have a nearly 40-year time period which we can use to analyze changes in lichen communities. So this is a really unique opportunity because lichen inventories are not as common as inventories of vascular plants, especially not over such a long time period. So this is what started raising the questions that would then form the foundation of my thesis. So these questions range from how does species composition in each quadrat change over time? And what trends can we see in species richness? Do we find that one growth form becomes more prevalent over the years? Do we see interspecies competition affecting these assemblages? And how does the nature of each individual quadrat change each of these trends, right? So this is a great way to start making inferences about succession in Sacchlids lichen communities in the flat tops. So in order to start answering these questions, I decided to return to the flat tops and add on a fourth time period to the inventories for the quadrats numbers 1, 2, and 3. So here we can see a map of the flat tops wilderness area. The 21 study sites established by Hale are designated by the yellow diamonds. And then the photo plots or quadrats are distributed amongst these sites. So I returned to site number 4, which contains the photo quadrats numbers 1, 2, and 3. So those quadrats are located on the rock surfaces in this talus field of volcanic boulders. So in the summer of 2019, I got to go with the Jabali lab to the flat tops, which was a wonderful experience. And I went and redid the inventories for each quadrat located in that talus field. So essentially, I located each quadrat, which was a task in and of itself, and using the species checklist from 2009, I looked to see if each species from that time period was present or absent in 2019. Additionally, I looked for novel species that had colonized the rock surface in the last 10 years. And for each species that I noted, I collected specimens in duplicate. Because with lichens, it's nearly impossible to identify two species accurately without doing a series of tests in the laboratory. You really need access to dissecting scopes and light microscopes and chemical tests to identify most species of lichens. So in order to do that, I collected these specimens, and we went out with hammers and chisels with a research collection permit to collect small portions of the rock containing each species that I hoped to confirm the identification of. So I took these species to both the University of Colorado Boulder and the U.S. National Herbarium. So I got to spend a week at the Smithsonian Institute with the help of a venture grant from Colorado College. And that enabled me to work in the basement there with all of their identification tools to identify my lichens. And then what was really cool is that I got to take my lichens and compare them to the lichens that Mason Hale collected in 1982 from the very same talus field. So that was an awesome opportunity to really validate my lichen identifications and then prepare my specimens for deposition. So when depositing specimens, you keep the substrate intact. So I put all of these rocks with the lichens attached into herbarium packets. And the labels for lichen specimens are very similar to the herbarium labels for vascular plants. So they include information about the species, who determines the identification, the locality, and the ecology of where they were collected, and so on. So now that I had all of my lichens identified, I was able to transition into analyzing these changes in the quadrats over time. So today I just want to take a moment to highlight some of the main conclusions from that portion of my research, beginning with our finding that there was a continued increase in species richness over time. So on the surface, this might seem like kind of an obvious conclusion, but there's actually a bit of debate as to what happens in a lichen community as the percent cover of lichens nears 100%. So if you imagine these quadrats, initially they were at one time, blank rock surfaces with lots of opportunity for lichen colonization, but now we're nearing a point where much of the rock surface is covered in lichens, so there's not a lot of space for new colonization. So there's two camps of thought on what happens to species richness when you hit this point. So the first camp says that lichen species richness will flatline or even begin to decrease once we hit this point where there's not a lot of bare rock to colonize. So the thought behind this is that the lichens that are already established will just begin to expand and assert dominance in this area, and there won't be a lot of opportunity for new colonization. So this theory has been predicted and supported by some field studies of Sacchlids lichen communities. But then there is this second ideology that species richness will continue to increase despite this decrease in available rock surface. So again, this been studied by several field studies, and our study of the flat tops supports this scenario, where we see this continued increase in species richness, even though there's not a lot of available rock surface. So how does this happen? And it's due to the continual senescence of lichen thalli on the rock surface. So as these individuals age, portions of their bodies start to crumble away, which then essentially creates a new surface for lichens to colonize. So we found out that the species richness in the flat tops wilderness area is really dynamic. It's a constantly ebbing and flowing process, but each apparent dieback actually just opens the door for further diversification. So you can see that here in this little chart of species richness. Let's take a look at quadrat number 1. You can see that dips in species richness in between 1992 and 2009. But then again, between 2009 and 2019, we already see it recover almost back to where it was. So that just proves that as one lichen dies out, it really opens the door for further diversification. Similarly, if we take a look at quadrat number 3, it experienced a net decrease of 4 species between 2009 and 2019. And that's the quadrat picture here. And what actually happened was a loss of 6 species and then the colonization of 2 additional species. So we're already seeing that the gaps created by these dying lichens are being refilled pretty quickly. So I would predict that if we kept revisiting this lichen community in the flat tops, we would see a continued upward trend with some dips in species richness that eventually rebound as colonization continues. So as you can see, this is still very early on in this study. We need a lot more decades to continue bolstering out this idea. But early on, the trend is definitely towards an increase in species richness over time. And when we look at the species composition of this quadrat, we see a notable increase in the abundance of foliose species over time. So in the 2019 field season, I discovered that there were 5 new species that had colonized the rock surfaces since 2009. And 4 out of these 5 species had the foliose growth form. So looking back to the foliose anatomy, what makes them a stronger competitor in a quadrat that's very limited by space? So if you think about it, a foliose lichen has some unique characteristics that enables it to compete in an area where there's not a lot of bare rock to colonize. And that's because it has this unique ability to grow over surfaces adjacent to it in a way that a crustose lichen cannot. So a foliose lichen, whether it has an umbilicate attachment, it may be able to grow over and extend across a neighboring lichen while still remaining attached to the bare rock at its umbilicus. Similarly, the rhizomes create this gap between the lower cortex of a lichen and then the substrate below it. So this gap is an opportunity to grow up and over a neighboring lichen. If you think about the anatomy of a crustose lichen in contrast, it grows so closely pressed to its surface below that it's directly lichen and then rock. There's no gap for which this lichen to overgrow its neighboring species. And you can actually see this process happening in quadrat 1, which is really cool to see kind of this theoretical thing happening in real time in this quadrat. So we see in the center, we have this white lichen in the years 1982 on the upper left, to 1992, then the lower left, 2009 and 2019. So this white lichen is Crustose, and it's neighbored to the left by a green foliose lichen. So you can see over the decades, this foliose lichen starts to encroach on the white lichen space. And in 2009, you can really see how it grows up and over the Crustose lichen. So this is a huge competitive advantage for this foliose lichen in a habitat that is limited by space. The Crustose lichen does not grow up and over the foliose lichen. It does not grow up and over the Crustose lichen to its right. So it's just at a competitive disadvantage. So it'd be really cool to track this exact quadrat over the coming decades to see if eventually the white Crustose lichen is entirely outcompeted by the foliose lichen. So again, that's part of the power of having these photographs at regular intervals, is you can track the competitive interactions of neighboring species. So there's just one more aspect of my research that I wanted to go over, and this is really where the community phylogenetics and evolutionary perspective come into play. So we were able to infer that the strongest interspecies competition was occurring on several of the horizontal quadrats in the Flat Tops Wilderness area. And so in order to infer interspecies competition, we looked to see how closely related the constituent species in each quadrat were. And the theory goes that the more closely related two species are, the stronger the competition between them will be. And so this is based on the notion that if two species are very closely related evolutionarily, they will have very similar ecological needs, and they will have very similar means of attaining these needs. And so if this competition is strong enough, then you will see a phenomenon called competitive exclusion occur. So when competitive exclusion happens, your two very closely related species compete eventually until one of them is excluded from the community. And so you can see evidence that competitive exclusion has occurred if you look at the phylogenetic structure of a lichen community. So in order to do this, we looked to see if the constituent species in each quadrat were more or less closely related than you would expect by chance. So to help illustrate this, here's a little mini phylogeny in which you can see that the closest relatives to each other have been excluded through interspecies competition. So in some of the quadrats of the Flat Tops Wilderness, we found that their phylogenetic structure indicated that closely related species had outcompeted each other, such that the surviving species were more distantly related than you would expect to see by chance. And so why did this happen in two quadrats, quadrant number 7 and 10, and not in the other quadrats? Why were these quadrats shaped by competitive exclusion while the others were not? And I inferred this to be because they are located on horizontal surfaces. So if you think about the biological requirements of a lichen, they need sunlight and they need water in the most basic sense. So the horizontal aspect allowed for adequate sunlight exposure. And what's really important is that a horizontal surface allows for water retention. So if you think about a vertical lichen surface where the rock is perhaps vertical or leaning over, when it rains, there's not going to be a lot of water retention on that surface. And since lichens do not have roots, they cannot simply absorb water up from their substrate. They really rely on a time period where their thallus remains wet so that they can absorb all of the water that they need for photosynthesis and to prevent desiccation. So a horizontal surface will allow the water to be retained on the surface long enough for the lichens to get the water that they need. So this is the last of the research conclusions I wanted to go over before going into some of the common lichens you can expect to find in Colorado. But if you are interested in more of the details or the methodology behind this work, then feel free to contact me. I'm more than happy to talk a little bit more about the nitty-gritty details of how community phylogenetics actually plays out. But the last thing I wanted to leave you all with today were some lichens that you could go out today into the field and recognize based on the morphological characteristics that we went over today. Because as a botanist and a biologist in general, I think one of my favorite things is just going on a hike and being able to point to things and say, oh, I know what that is, or I know the function of that structure. I can recognize the organism that I'm looking at. So I wanted to leave you all with a couple of species and genera that you can do that with today. So the first is Rhizocton geographicum. This is the map lichen. So it's very common in Colorado. It's a crustose species with a distinctly bright green to bright yellow thallus. So it is an areolate lichen. So it has these little scales that are angular in shape. If you look at those little yellow sections comprising the thallus, they are not round. And it also has a conspicuous black prothallus, which is that fungal mat that I had mentioned earlier. So in this case, you can see the prothallus present around this individual, and you can see that there are actually 3 individuals of Rhizocton geographicum present in this image. Looking at the reproductive structures, this lichen has Apothecia, which are black, and similar to the areoles, they are angular in shape. And these are Lecidine Apothecia. So they do not have a ring of yellow lichenized tissue around them. They are just simply the black Apothecia as themselves. So this lichen is a Sacchlids lichen. You'll never find it growing on trees or on soil, simply on rock surfaces. And it's very common in coniferous regions between 3,000 and 12,000 feet in altitude. It has a preference for south-facing slopes, and it's commonly found in later stages of succession. So this is a very common lichen, you're likely to come across in Colorado. Next, we'll take a look at my personal favorite lichen in the state, which is Rhizoplaca chrysaluca. It's a foliose lichen with an umbilicate attachment, and it has this whitish gray or greenish yellow thallus. So in these 2 images, you can see how the thallus can vary in color, and its underside is tan to dark brown. So its apothecia, which are my favorite part about it, are very abundant. Oftentimes the surface will be just covered in apothecia, and they're lechinorine. So you can see that ring of lichenized tissue surrounding them. They're this beautiful peachy orange salmon color, and they are often pruinose. So pruina is simply a dusting that covers portions of the lichen that can be used to help identify them. It makes them kind of whitish gray in appearance. So this lichen is, again, Sacchlids. It only grows on rocks, and it's common in pinyon juniper woodlands to alpine. Next, we have the genus Clidonia, the pixie cup lichens, which is a really fun lichen to come across. And like I mentioned earlier, Clidonia is unique in that it has both a primary thallus and a secondary thallus. So the primary thallus is composed of squamules, which are sometimes steridiate. And many of the species in Colorado have this green to brown primary thallus. The secondary thallus is the upright fruticose portion, and those structures are called podetia. And they have either blunt, pointed, or cup-shaped tips. You can see the cup-shaped tips in these images here, and they may be branched as well. So the apothecia of this lichen are located at the tips of the podetia, and they are lecidine as well, so they lack that ring around them. And they are a dark red to brown in color. So these lichens, you'll commonly find them growing on soil, rotting wood, or the bases of tree trunks. They also grow out of moss occasionally. So we have just one more genus I'd like to go over today before we open it up to questions, and that is Usmia, the beard lichens. So this is a fruticose lichen with a central holdfast, and it can either be shrubby to long and pendant in nature, and it will have that round cross-section since its thallus is cylindrical. So it's really common to have this frosty green to yellowish appearance, and what's unique about Usmia is that it has a central elastic cartilaginous cord. So if you take one of those branches and pull it apart, you'll find that the center is elastic, and that's really distinctive of this genus. So its apothecia are lechinorine. They have a ring of lichenized thallus around them, and they may be pruinose. So this species is really commonly found on bark, wood, and rocks, especially on coniferous trees in Colorado. If you like where with conifers, you will likely see this growing on the twigs of those trees. So this rounds out that portion of my talk. I just wanted to go ahead and leave you all with some resources and further reading if you're interested in learning more about lichens. These were all tools that were really instrumental to me being able to learn how to identify and recognize lichen features in the field. So feel free to keep these in mind if you're interested in learning some more about lichens. And now we will go ahead and open it back up to questions.
Piper, so I can read the chat and tell you what folks have been talking about. And we'll start there, and then people get brave, write more questions. Firstly, thank you so much. That was an excellent talk. I learned a lot. And I appreciated your commentary on ecology. So we have one comment. We were talking about how the Fruticos lichen overtakes the Crustose lichen. And do you think this is a form of succession?
I think it's a pattern that is often observed along the successional process. So as I said, I think this is just something that makes sense in terms of competition, because we see in lichen communities that competition kind of ebbs and flows, right? So if you have that bare rock initially, there's not going to be a lot of competition between lichens, but you're going to have a really harsh environment that's hard to colonize initially. So in the early stages of succession, it's kind of everyone's game in terms of colonizing that surface right off the bat. But as you start to add in more species, that's when the specific characteristics of each individual lichen really come into play in terms of competition. So I think competition is really mediated by successional stages. And it's interesting because in some epiphytic lichens that are growing on tree trunks, we actually see the opposite. So I read a study where the tree trunks usually had foliose species earlier on in succession, but then they transitioned to crustose species. So that's an interesting change depending on your substrate. So I think it's an area that needs to be studied more, but as far as Sacchlids lichens go, I do think there's a link between succession and growth form.
Okay, thank you. So we also were wondering, do you get a chance to do any element or chemical analysis to follow up on earlier work to assess the air quality impacts of that longer study you were talking about?
Right, so I did not do any of that work on my own, but the cool thing about depositing specimens into herbaria is that both in the University of Colorado Boulder Herbarium and the US National Herbarium, now there are like these little time capsules to the summer of 2019. So these lichens, even though I didn't perform the analysis at the time, the information is still there. So if anyone is interested in continuing that part of the study, then my lichens are available for that, and you can still get the same information even though I didn't do it at the time. Yeah.
Thank you. That's an important point. We also, as an organization, have been working on the curation of iNaturalist, thinking about what we have to verify species there. And so I was wondering, are lichens in like SciNet and these global diversity index, etc.?
Yeah, and that's a great question. So actually, there is a lichen version of SciNet, which is the Consortium of North American Lichen Herbaria, which is an incredible tool. So it has pretty much all the same features that SciNet has. You can do specimen searches, and it has just tons of information and digitized lichen herbarium labels and specimens. So that was a really instrumental resource for me doing my lichen research. They have species checklists for lots of different areas. So for instance, I think Dinosaur National Monument has an entire species checklist for all the lichens known there. They're working on one for Rocky Mountain National Park. And this is part of a ongoing grant right now that is expanding this North American consortium to a global level. So it's a really good resource that if you're interested in looking into lichen characteristics, lichen collections, maps of where lichens have been collected, all of that information is available on that database.
Thank you. And do different lichens prefer certain host rocks?
Yeah, so there's some species that are more generalist. So some species you can find growing on rocks and trees and bark, but then there are some species that are really specific in their ecological requirements. So for instance, some lichens will only grow on a certain type of rock because it has the acidity requirements for it, it has the right texture for the lichen, it has the right water retention characteristics for that lichen. So there's definitely rock specificity. in certain lichens. Generally, sandstone is a less diverse lichen substrate. Not a lot of lichens prefer sandstone for whatever reason. But then looking at volcanic rocks, then you often see a greater diversity of lichens growing there. So there's definitely host specificity when it comes to rocks.
All right. And would you like to take a moment to explain asexual reproduction in lichens?
Absolutely, yeah. So asexual reproduction in lichens is really important, as I said, because sexual reproduction is not a very guaranteed means of reproduction. So asexual reproduction involves the production of propagules. So these are simply just portions of the lichen thallus that are then dispersed out into the environment. So there's no, sexual variety taking place here. It's simply the same exact individual with the same exact genetic makeup being dispersed out into the environment. But the lichen does this in a variety of different ways. It can be as simple as fragmenting the lichen in half, and then that newly broken off piece can go adhere to another substrate and carry on as a new individual. But lichens have also evolved this ability to produce actual structures designed for asexual reproduction. So these include the Siridia that I mentioned, which essentially are just a few algal cells surrounded by a layer of fungal tissue. So what's really important is that you're dispersing the photobiont and the mycobiont together. That's the key part of asexual reproduction. So by combining those two features and producing them in either spheres like Siridia, and then as little cylinders as Isidia, those can then break off and start new lichen individuals once they break off the surface of the lichen. And does that answer that question?
Hopefully that person will write in the chat.
Yes, hopefully that is sufficient. If you have any further questions on that process, I'm happy to talk more about it.
Yeah, well, we can relate to that as botanists, looking at floral species, because vegetative growth is one way to spread a species around. And certain of our non-natives are very good at this, like fragments of bindweed is what I was thinking of inside the soil. Right. They can just be transported around and become new organisms. Okay. Judith? Oh, I thought someone was going to say something. Okay. And now, here we have the next question. How about using lichens for age dating?
Yeah, so I actually did a quick presentation on this in one of my anthropology classes, but lichens are a really cool tool for doing age dating as well. So essentially, if you have a lichen growing on an archaeological artifact and you know the growth rate of that lichen, then you're able to determine the age of that artifact. So that's a really cool application that's been used to date some cairns in Colorado. I know that's some work that's been done here. Also, certain glacial events have been dated by analyzing the lichens on those surfaces, because as long as you know the growth rate of that lichen, then you can do calculations to see how long ago that rock surface was exposed, and you assume that the lichen began colonizing that surface at the time that that was exposed. So that's a great application of studying the size of lichens and their growth rates to date surfaces.
Okay, now we have from Russ, great talk. I understand you're located in Phoenix. If you do make it down to Southwest New Mexico now and then, it'd be fun to do some lichen ID here in the Gila National Forest. Larry's saying Claire has done quite a bit of work here, and the email is supplied. So that's awesome. You're down in Arizona, you can get there.
Yeah, that sounds wonderful.
And now a question about collecting specimens. If one does legally collect specimens, how long do they remain viable for growth? Can they be grown in home garden if you can provide the same substrate? Are there resources to discuss growing them at home? And then the pre-question would be, what are the rules about collecting lichens?
Right, yeah, so for instance, for my collections, I had to work with the Forest Service to obtain a research collection permit. So definitely depending on where you're planning on collecting, there's different permitting processes involved. From just lichens, you know, if you find them in your backyard, there's definitely potential for that. And the ones I've kind of collected for my personal collection, I have some that are hanging out on rocks around my apartment, but in terms of actually growing them, it's not like a plant. It's not like, oh, water it, you know, every so often and give it this amount of sunlight. It's a little more complicated than that. There's not, as far as I know, too many resources about gardening lichens, but it'll be a very cool kind of avenue to look more into, but I'm not too familiar with actually trying to grow them.
Okay, now how about where would we find a list or some information that would link lichens to specific rock types? And then a follow-up question, is the growth rate constant, and how do we know?
Right, okay, so first, I would recommend that Consortium of North American Lichen Herbaria. It's a really good resource if you know the species you're looking at and you want to know what rock types it grows on. It has this category where in its description it lists the different substrates that this lichen is known to grow on. So that's a good resource for all of that. Additionally, the Lichens of North America, that is this textbook on the right side of the screen, that is kind of the holy grail for everything lichen in North America. So for every species described in that book, it gives a description of the substrates it's known to grow on. So I think those are really the best resources if you want to learn more about the ecology of a specific species. And then as far as the growth rate, so the growth rate isn't necessarily constant over time, but one of the best ways to establish growth rate is to look at a cemetery. So this is actually how a lot of times lichen is used to help date structures, is you go to a cemetery that's located in a similar ecological condition. And if you look at a gravestone and you know how long it's been there and you know the size of the lichen, you can start to assemble a series of different lichens of different ages and see what sizes they are. So say you collect data on the same species of lichen from 50 different gravestones in one cemetery, and you can start to kind of average out their typical growth rate using a method like that. Mm-hmm.
Wow, that's cool.
Yeah, really cool application. Mm-hmm.
Oh man, and I've done labs in cemeteries, but not ones like that. That would be a really fun lab.
Yeah, absolutely.
We don't have any other questions, although you did an awesome job at answering those on the fly. So I think we're at 11:04 and we have a little bit more time. But essentially we want to say thank you for joining us.